Hey guys, welcome to another A-level biology video. And this video will be about the topic control and coordination in plants. So, plants, like animals, need to be able to respond to external and internal stimuli, and for that they need some system of communication. So most plant responses involve changing some aspect of their growth to respond to factors such as gravity, light, and water availability. And plants are also able to quickly respond to changes in CO2 concentration, lack of water, grazing by animals, and infection by fungi and bacteria. Right? So basically, their communication system is pretty reasonably strong, and you might not be already aware of this. You may not have thought about this, but you will get an idea of this in this video. So, some of these responses are brought about by quick changes in turgidity, right? As happens when stomata respond to changes in humidity, CO2 concentration, and water availability. And another example is of the mimosa plant. If you were to touch the mimosa plant, leaves would quickly roll up, and this change is actually brought about by changes in the turgidity. While plants don't have the sort of complex nervous system that mammals do, they do have something that resembles electrical communication. There are differences though in the electrical communication that you would find in a mammal and within a plant. There are similarities and there are differences. Similarities, that electrochemical gradients do exist between the cytoplasm of the cells and the immediate surroundings. And action potentials do get triggered, and repolarization is indeed taking place. So some of the basic aspects of neuronal communication is there. But the differences are that depolarization is not achieved in the same manner as in mammals. What is depolarization? Depolarization is basically changes in the electrochemical gradient or differences in the distribution of electrical charge across a cell surface membrane, right? So that change is the creation of a positive, or the creation of an electrochemical gradient with greater positivity in the cytoplasm compared to the outside of the cell on the other side of the cell surface membrane. While in mammals, this depolarization is achieved by the influx of Na+ such that there are more positive charges inside than outside. But in plants, this depolarization or this creation of positivity is actually achieved by the outflow of Cl-. So when there is less negativity within the cytoplasm, there would be more positivity, right? But either way, and the same result, the same sort of depolarization is achieved. The difference is that there aren't any actual nerve cells or any specialized nerve cells found in plants like there are in mammals. So how do action potentials travel? Because we do say that action potentials are indeed triggered. Well, action potentials travel along the cell membrane from cell to cell via plasmodesmata. So plasmodesmata play a huge role in transmission of action potentials, right? Because cell walls come in the way, so direct transmission doesn't really seem to be a possibility, right? But at the points of plasmodesmata, we don't have cell walls. So transmission is a possibility. And the last difference is that the action potentials that are indeed triggered in plant cells last longer and they travel more slowly, which makes sense because we don't have any of that specialized nerve cell. We don't have myelin sheaths, which greatly increase the rate of transmission, and so they last longer and they also travel slowly. So you can say that some but not all plants are capable of producing action potentials, and in the examples you can write the mimosa plant. So microelectrodes, if they're inserted into leaf cells, detect changes in potential difference that are very similar to action potentials in animals. However, the depolarization results not from the influx of positively charged sodium ions but from the outflow of negatively charged chloride ions. Repolarization, on the other hand, is achieved in the same manner by the outflow of potassium ions. So, both depolarization and repolarization happen, but whereas repolarization happens in the same way, depolarization is achieved in a slightly different manner, but with the same end result. So plants do not have specific nerve cells, but many of their cells transmit waves of electrical activity that are very similar to those transmitted along the neurons of animals. and the action potentials travel along the cell membranes of plant cells and from cell to cell. So the action potentials travel along the cell membranes of plant cells and from cell to cell through plasmodesmata that are lined by cell membrane. So the action potentials generally last much longer and travel more slowly than in animal neurons. So many different stimuli can trigger action potentials in plants. For example, chemicals coming into contact with a plant surface may trigger action potential. For example, if you drip a solution of acid or a similar pH to acid rain on soybean, action potentials will sweep across the soybeans in potato plants. Colorado beetle larvae feeding on leaves have been shown to induce action potentials. No one knows what effect, if any, these action potentials have, but it is thought that they might bring about changes in the metabolic reactions taking place in some parts of the plant. So, this is a branch of biology which we don't know a lot about. We do know that in certain circumstances, action potentials are indeed triggered to external stimuli, but we aren't sure what exactly those action potentials manage to achieve. We theorize that they may alter metabolic reactions, but we are not sure. As of right now, this is merely a hypothesis. We are merely presuming. And now they're going to mention the Venus flytrap, and this is actually mentioned in the syllabus, I believe. So I would suggest we really get to it. So the thing about plant cells is that, right, so the learning outcome is plant coordination systems involve rapid responses, as in the case of the Venus flytrap, but also complex interactions between plant growth regulators such as auxin and gibberellin, and plants respond quite differently to different concentrations of plant growth regulators. So the concentration actually determines what the response will be. And the first learning outcome is describe the rapid response of the Venus flytrap to stimulation of hairs on the lobes of modified leaves and explain how the closure of the trap is achieved. So this is actually doesn't seem all that complicated, but there are a lot of steps. So this is lobe 1 and this is lobe 2 and this is the midrib. So the specialized leaf is divided into 2 lobes either side of a midrib. This is the midrib, right? And the inside of each lobe is often red. So this is all red, and there's nectar secreting glands around the edge. So there would be nectar secreting glands dotted around the edges, and the purpose is to sort of like attract insects, because otherwise they don't really have any sort of incentive to go sit on the big red looking scary leaf, right? And each lobe has 3 stiff sensory hairs. Alright, so these sensory hairs are basically very stiff, and then they aren't like our hair which just blew away, meaning they're really stiff. And by sensory hair you can see that they're definitely some sort of a receptor. So 3 stiff sensory hair per lobe, right? And they're the ones that respond if they're deflected, meaning if an insect comes along and sort of like bumps into one of them, sort of like deflects them, the deflection will be I don't know what to call it, deflection would be felt, right? Now the outer edges, this is a bit too thick, isn't it? And so the outer edges have these closely located stiff hairs, and they're all over, and they're all the way over each lobe. And look something, or they're arranged like this. And the same way on the other side of the lobe, and on the surface of the lobe, as in over here. So on the surface of the lobes there are many other kind of glands dotted, and they aren't like the nectar glands that are on the outer edge. These glands actually secrete digestive enzymes. So if a fly or some other insect were to come on and sit on the lobe and bump into the sensory hairs that are on the inside of the folded leaves, and these one, not these ones, these are stiff but these aren't sensory, and if they were to sort of bump into these, the venous flytrap would be stimulated because of action potentials which will travel very fast across the leaf, and those action potentials will cause it to fold over and trap the insect. But it's not quite so simple. So what does this initial deflection do? Well, this might be taking place in plants, but the stiff cells are still sort of a receptor, and so they operate in the same way that receptors in mammals do. So when a taste bud or the specialized cells of a taste bud are stimulated, calcium ions are actually stimulated to enter, and this causes the creation of a receptor potential. And if this receptor potential is high enough, an action potential will be generated in the receptor neuron that is associated with the receptor. So something similar happens. So if a sensory here is deflected, the deflection activates calcium ion channels that are at the base. So there are calcium ion channels that are at the base of these stiff sensory hairs, and these channels then open because of the deflection, because of the insect bumping into them. And so calcium basically flows in, and this flow generates something called a receptor potential. Now, if just one of these hairs is stimulated and the insect just stands over there for a while and then maybe flies away, then the venous trap won't close. For a venous trap to close, two of these sensory hairs or one of these has to be stimulated twice within a period of 20 to 35 seconds. And only then will action potentials travel across the trap. So when the second trigger takes too long to occur after the first, the trap will not close and a new time interval starts again. So if one hair is triggered, but the insect just being all lazy doesn't trigger another one, and 40 seconds pass and then it triggers another sensory hair, then the trap won't close, but another time period will start. So if say it triggers a third cell in 15 seconds, then the venous trap will close up. So the time between the stimulus and the response is about 0.5 seconds, and it takes the trap less than 0.3 seconds to close and trap the insect. So basically, if after triggering the second sensory hair, if the insect is really quick and somehow manages to escape or leave the loop within 0.8 seconds, then it's home free, but it likely won't be that quick and it will get trapped, because after stimulation of the second cell, it takes 0.5 seconds for the action potentials to travel across the loop, and a further 0.3 seconds for the trap to respond by closing and trapping the insect. Rapidly changes into a concave shape, bending downwards, so the trap snaps shut. So this is convex, and so this will be concave. So that's the sort of shape it gets. And this sudden change in shape is what causes this trap to snapshot. And why do we theorize that this has to do with action potentials and something a bit more complicated than just mere changes in turgidities? Because this happens way too fast. It just takes 0.8 seconds. Changes in turgidity tend to take longer. And this happens too fast to be simply the result of water movement from cells on the top of the lobes to cells underneath. And instead it is likely that the rapid change occurs as a result of a release of elastic tension in the cell walls. So they're saying that this is, so some might have theorized at some point that this change, the fact that this was sort of like really bulging upwards, might have been due to the fact that water would have left the cells that are right at the top and gone downwards, and because the cells would have lost their rigidity, they would no longer be swollen, and so the lobe would not be bulging upwards any longer. But this wasn't really all that well received because the change was way too quick for to be due to turgidity alone or changes in turgidity alone. So what they theorized was that it was likely that the cells at the top, their cell walls were in a state of elastic tension, right? So they were really tense, like when you pull on a rope, it's really tense. But when you let go of the elastic tension, it sort of like coils back into its original shape. So when the elastic tension was released, the cells sort of like didn't bulge up anymore. However, after the initial interlocking,